Okay. All right. So let's go on. So now this one, it's sort of this part of the talk is kind of where the rubber meets the road, where you actually have to get down to brass tacks, really geeky things about estimating effective size. And I got to say, it's kind of interesting to find so any geeks who are interested in this topic to talk to. So that anyway, that's what I'm talking about here. Here's some more hyperbole about effective size. Here's this beautiful kind of poetic quote by Richard Feynman, motivated by his work on physics or so on. But he's talking about nature uses only the longest threads in the tapestry of life and or nature. And again, Fred Allendorf has sort of taken off in this and claims that effective size is a primary thread in the tapestry, population genetics. So more poetry. But it raises a question, can we get carried away here a little bit? So is there a worry that we can be too obsessed with focusing on effective size? I mean, this isn't really a scientific question to answer this, but there are a couple observations. It's certainly the case that a lot of people worship a lot of things sillier than effective population size. And it's also true that definitely, I'd say pretty much incontrovertible, that NE is really important. So here, I did just as exercise, I submitted to Google this statement or partial statement. One of the most important parameters in evolutionary biology is, and Google's AI came back saying it's effective population size. So you got to give it credit there, but I only give it about a C for this. You only got it half right, because look what it says. The next, it should have stopped when it was ahead. So this is fake news, this part in red. It's just found this false statement somewhere in the online universe and regurgitating it in its AI overview. So, okay, for more seriously, important reference point for considering effective population size, it's what's called a Wright-Fisher ideal population. And this population has really specialized characteristics. It's got discrete generations, no migration or selection, or mutation, actually. It's got an equal, if it's got separate sexes, they have to be an equal sex ratio, got random mating, and every individual has to have equal expected reproductive success. And in these populations, then, on average, you're going to get this Poisson variance in offspring number, and on average, you're going to get effective size equal with census size. Okay, so that's kind of repeating what we just talked about earlier. Well, there are lots of deviations from that. One of them, that is, most species are not discrete generations. Most species are age structured. And for these species, we have to consider two kinds of effective size. We still have NE per generation because all of the population genetics theory, almost all, certainly originally developed assumes discrete generations. And so if you want to relate your results to standard population genetics theory, you have to be talking about effective size per generation. But any Heteropara species, if you're going to collect data for Heteropara species, you're going to collect data every year or season, and then add them up across lifetimes to get something related to whole generation or lifetime. So that raises a question, well, what about this NB parameter, which is the effective number of parents that reproduce in 1 year? Actually, this term NB is a result of help from Joe Felsenstein. I was trying to figure out what happens in Pacific salmon, who a lot of like Chinook salmon may mature anywhere from 3 to 6 or 7 years old. But each year, all the spawners die after spawning. So people at the time, even people who were leading geneticists at the time, were measuring reproductive success in 1 year and calling it NE. Well, I knew it wasn't NE, but I didn't know what it was. So I explained this to Joe Felsenstein. And he said, oh, well, that would be the effective number of breeders. And so that's the origin of the term NB. I use sub B instead of NE to talk about effective breeders. But it's also true for heteropara species that reproduce multiple times. So this is a useful parameter because it reflects evolutionary processes going on at one point in time. For example, sexual selection. That occurs in 1 year or a season. Those are the individuals you're competing with, for example. So both of these are important, but for different reasons. So NB per year can be calculated using the discrete generation formula for inbreeding NE, which is not affected by the offspring number. That variation in offspring, mean offspring number is problematical for demographic interpretation of variance. And that's why inbreeding NE formula is most useful for looking at NB. So this just took the formula for inbreeding NE for discrete generations and turned it into a ratio. And so again, you have this ratio of variance to mean offspring number, this time in just within 1 year. That's really important. So what are the components for variation in offspring number per year? Well, there's an among age effect. This is reflected in the age-specific fecundity term, or BX, in a standard life table. Does BX change with age? If so, then you have the fact that individuals of different ages have different expectations of reproductive success. That's going to increase VK. Within age effect, you're interested in what's the ratio of variance in offspring number to mean offspring number for individuals the same age and sex, like 6-year-old males. You know from a life table what the average reproductive success is for 6-year-old males, but what's the variance? That's a key parameter here. You don't know what VK or NB is if you don't know that, but people almost never report it. Even in life tables, if they have data to calculate a mean, they must have data to calculate a variance. But unfortunately, that parameter is, it's really hard to get empirical data on that parameter. Relationship between NB and NE, really important if you can go between one and the other, because sometimes it's easier to estimate one than the other. Turns out in simulpara species that are age structured, this includes Pacific salmon and also annual plants with seed banks. It turns out effective size is largely additive. You have 100% turnover of the individuals reproducing each year. So effective size is kind of additive across the years in a generation. So G there is generation. Heteropara species, the ratio of NB to NE actually varies a lot and depends on some key life history traits. So that's kind of an interesting result, but it's generally more similar. It's not as much, NB is generally not as much smaller than NE as it is in the simulpara species. How do you calculate NE directly? With demographic data, the most widely used method is by Bill Hill, or again, about 50 years ago. And this simplifies his formula a bit, but basically the NE-to-N ratio here in these iteropair species is roughly proportional of 4 times the generation rate over 2 plus, in this case, lifetime variance in offspring number. Okay, so the asterisk here means you have to add up reproductive success of individuals across every year of their lifetime. Understand, not surprisingly, that's really hard parameter to estimate or collect empirical data for, particularly for long-lived species. There are quite a few other factors that affect this lifetime variance in reproductive success. So one is persistent individual differences. So a null hypothesis might be that, okay, 1 year, some individuals just by chance are going to reproduce more offspring than others, but next year, are the same ones likely to be good producers, or are they just as likely to be underperformers next year? That'd be an independent null hypothesis. But in fact, in the real world, there are a lot of times where some individuals just consistently are really good or really bad at reproducing. So this is called persistent individual differences. This is work done here in Trondheim, and it has no effect on NB, but it reduces effective size because it increases the lifetime variance in reproductive success. The same individuals consistently being either good or bad. Skip breeding has the opposite effect. It's sort of intermittent breeding, common in many species, particularly females may skip one or more reproductive seasons after energetically costly reproduction. This reduces ND because there are fewer available breeders, but it increases effective size because it limits the ability of certain individuals to always reproduce, to be top reproducers every year. So it's a modulating effect, actually. Small litter size, that's common in many species, particularly large mammals. A lot of females in whales or elephants or so on. Basically, the only options for a female in those years are 0 or 1. And if the only options for reproduction are 0 or 1, it's easy to show it's impossible to have the variance even as large as the mean. Variance will always be less than the mean. So this means that, again, you're constraining how large the variance can be, both across 1 year and across a lifetime. So several factors there that have important influence on lifetime reproductive success and NE. Now we're going to talk about demographic estimates of effective size. Those what I talked about so far are demographic true effective sizes. Major sources of uncertainty for demographic estimate of effective size then are first, uncertainty in pedigree reconstruction. This is if you have uncertainty how many offspring every individual produced. And the degree to which that's true or a problem will vary considerably depending on the study. But in fact, parentage analysis generally does not require dramatically large amounts of genetic data in most cases. So if you just want to get parent offspring data for getting reproductive success, it's generally pretty feasible to get pretty reliable data. The other issue is sampling of parents and offspring. So this is probably overall a bigger contribution to uncertainty. So just a couple generic scenarios here with respect to the second point. One scenario might be, let's say you have identified all potential parents, genotype them, say, and you have a random sample of the offspring, but not all of the offspring. In that case, your estimate of inbreeding effective size is not biased. If you use inbreeding, that wouldn't be true if you use the variance in the formula. but precision is going to be greater if you have larger samples of offspring. So it's not an issue of bias, just precision. Now consider a scenario where you have random samples of both parents and offspring. In that case, your estimate of effective size, either NE or NB, is going to be biased downwards. You don't have any data for some subset of parents. But if both parents and offspring are randomly sampled, your estimate of NE over N or NB over N will not be biased. Okay, so those are just examples of some ways the uncertainty plays out in estimating demographics. The rest of the time, I want to talk about genetic estimates, effective size, become very popular in recent years. And what happened was that this is basically the same graph I showed you before that shows the inverse relationship between genetic drift and effective population size. And at some point, somebody realized that, well, if you can take effective size and predict from this and this curve, the rate of genetic drift, if you could measure or estimate the rate of genetic drift, you could work backwards, reverse this process, and use that information here to estimate effective size. So that's the whole basis for this field of depth. So these genetic methods, they can provide potentially information about historically any contemporary effective size, which basically is effective size that applies to the period spanned by your samples, say, or bottlenecks, rapid changes in effective size. And just because we got limited time, there's lots of examples in the literature of all of this. We're going to focus on estimating contemporary NE. That's also most directly relevant to the issues folks here are talking about. There are more methods in the literature you can find, but the three main ones that I think by far have been used the most: the two-sample method for estimating effective size based on temporal changes in allele frequency. The temporal method requires at least two samples. And two one-sample methods that estimate inbreeding effective size in the parental generation. And these are based on linkage disequilibrium, which is random associates in alleles at different loci, and the SIP-SHIP method of Jin-Liang Wang. Okay, so those are the three that are most common use. It's a quick explanation of how the LD method works. The temporal method is pretty comparable in form, but basically, from theory, is your expectation of this R-squared statistic, which is a squared correlation of alleles at different loci. It's easy to compute from genomics data or genetic data, and that has two components to expectation. There's a drift signal that comes from here in this case, 1/3 times NE, and a random noise effect that's associated with the fact that you're sampling a finite number of individuals. Here, S individuals. So you can see this random noise gets smaller the more individuals you sample. So that's a good way to increase precision, increase your. So this also shows the drift signal is going to be strongest when NE is small. So estimated effective size is much easier when effective size is small. When effective size is really large, this term becomes really small, and it's hard to distinguish from the noise. So it's easy to just turn this equation around to get an estimator of NE here. And generally then what is done is you compute the raw R-squared value here. You subtract the average amount expected to be due to sampling error. and then use that to estimate effective size, because what's left after accounting for sampling error should only be due to genetic drift in theory. One of the problems is that, particularly if NE is large, this, after you subtract this expected amount from sampling error, you may actually end up with a negative number. And that is then has to be interpreted as an effective size estimate of infinity, because you got no evidence that genetic drift was involved. Because all of the LD you found, for example, can be explained by sampling error alone in that case. Temporal method has very similar structure, except this term then has a term for number of generations in the numerator and any in the denominator. For the LD method, when there's linkage, this applies to alleles on different chromosomes that are physically unlinked and assorted independent. If there's physical linkage, so you have comparing alleles from different loci on the same chromosome, you still can estimate effective size, but it also depends on the recombination fraction between the genes. And that's the whole basis for methods. Some of the methods that look back in time to estimate effective size and get a temporal trends of effective size from methods like this that use physical linkage. And the reason you can do that is because for tightly linked loci contain information about effective size farther back in time. The SIP-SHIP method considers three types of relationship in a single generation pedigree, so parents and offspring. Full SIP, you're comparing pairs of individuals, okay, that are offspring, and they're either then put in three bins: full siblings, half siblings, and unrelated. And here the unrelated, only with respect to this most recent generation. It could be cousins or something else more distant related, but that's not relevant to estimating you're estimating effective size just in the parental generation. And then these are half sibling and full sibling doses in the denominator. So once you add up all the SIP-SHIPs in your samples, it's easy to estimate effective size. So Wang's method described in 2009 has been also widely used. If there are overlapping generations, and if you have samples from a single cohort, that again, like the LD method, would apply to NB, the parents reproducing in a single year. Now, these genetic methods have a lot of assumptions, and there are lots of ways they can be potentially biased if these assumptions aren't met. There's quite a bit of literature evaluating performance of the different methods under different scenarios. So you can look in some of the cited papers to find that. I'm going to talk just about a few of those issues. One is, this doesn't have to do with bias, but it has to do with precision. Why is estimating large NE so hard? Well, it's a signal to noise issue problem. So here's on the top is a pretty easy problem. You got a situation where you got sample size of 50 individuals and true NE is 100, so the drift signal is pretty big. We got a temporal method with 3 generations of drift, so 3 times the drift signal here. So this is the overall drift signal, and you've got a lot of sampling error, but if you get enough loci, you can estimate both of these parameters pretty carefully and basically subtract out this, and you're left with this pretty strong drift signal. Here's a situation you have the same amount of data, but true NE is 10,000. This is the relative size of the drift signal in black to the noise. It's almost like a needle in a haystack kind of point. So in a nutshell, that's why it's so hard to estimate true NE in really large populations. This, I'm going to skip this. It explains the same principle, a little more complicated ways. So for genetic estimates of effective size, there's two major sources of uncertainty. You have sampling genes and you have sampling individuals. Genes, say in a typical vertebrate population, might have 10 to the 9th or 10 to the 10th DNA base pairs in a genome. And even a big genomics data set is probably going to have no more than a few thousand or maybe up to 1 million or so SNPs for a non-model species. So you're still sampling just a small fraction of all the genes. And sampling individuals, you're sampling just probably some of the offspring potential reconstructing pedigree. And the important point here is that whatever answer you get is going to be very sensitive to the pedigree of the individuals who appear in your sample. But generally, what you want to draw, you don't care specifically in general about the pedigree of the individuals in your sample. You want to draw inference about evolutionary consequences of the pedigree of the population as a whole. So even if the sampling is random, it can be not representative of the pedigree as a whole. Here's an example that looks at the relative value in experimental design terms of the value of loci versus individuals. So back in the old days where you say you had just a handful or 10 or 20 or 30, say, allozymes or microsatellites, they ask the question, if you want to double the number of loci you have or double the number of individuals you have, here we start with sample of 25 individuals and 50 loci, 50 SNPs, I think is here. And if you held one constant and doubled the other, if you did it with doubling loci, you could reduce, this is the variance of R squared in this case, reduction in variance of R squared, which result then in an increase in precision in your estimate of effective size. And you can see you got about the same reduction in the variance from doubling individuals and the whole number of loci. So in the old days, roughly equal for experimental design purposes. But that's not true anymore in genomic scale data sets. So this is looking at the same sort of scenario. Whoop. Yeah, this is why you're not going to get the same result, because you have to deal with this issue in genomic scale data sets, this law of diminishing marginal utility, which is once you have a certain number of a certain thing, the value of one more of those items to consume is generally less than you had before. So this just shows what happens for drinking beers. First beer tastes really good, that's high value. The next one on a hot day might be almost as good, but by the time they're out to the second six pack, the added value of another beer really going down fast. For the relevance for genomic scale data sets is the following. The average number of chromosomes in these major groups is somewhere around 10 to 20 for most higher organisms. So if you're talking about thousands or even millions of SNPs having to be crammed into this small number of chromosomes, at some point, you're going to find a situation where you add one more locus. Where are you going to put it and get it far enough away from existing loci that it's going to provide any useful information that's not already captured by the data you have? It gets increasingly hard because the closer it is to any existing loci, the less useful the information will be. It'll just be the same information multiple times. So that's the basic problem. What that means is this shows the marginal utility in adding 10 loci, I think, for just estimation FST and for estimation LD NE with the LD method. And you can see that, so this would be the pattern. It should be similar to estimating temporal depth here. It's encouraging for the temporal method in that even though the marginal utility is going down, it's not too dramatic. So even at 50,000 SNPs, each new SNP you add isn't as valuable as the first ones were, but it's almost half as valuable. So you're still getting a lot more information. So you can use genomic scale data sets pretty effectively with the temporal method. That's not true of the LD method. The LD method starts really rapid increases because the number of pairs of loci you can use goes up with the square of the number of loci. So you see a huge increase in the marginal utility of adding more loci, but then it drops rapidly. So by the time you've got to 500 or 1,000 loci, you pretty much milk the amount of information you're going to get from the LD method. The marginal increases for increasing loci are pretty small after a few thousand loci. You can see there's a 10-fold difference in the x-axis scales of these two here. So it's really dramatic difference. So really large genomic scale data sets are beneficial for the temporal method compared to the LD method. So here is going back to this initial thing with doubling loci or individuals. For the LD method, now we're looking at a different situation. When you start out with already have 5,000 loci in that, and you still have only 25 individuals. Well, if you double the number of loci and hold the number of individuals constant, now you got 10,000 loci. You've hardly had any reduction, further reduction in the variance of R squared at all, just because you pretty much, even with 5,000 loci, you pretty much maxed out the information content. But if you double the number, stay at 5,000 loci and double the number of individuals, you got a huge increase in precision. Okay, so what this means is, we scale data sets, individuals are relatively more valuable than loci. Loci can become largely expendable. So you can really think about, you should really think about that in your filtering process. You have, in filtering, you can have some options for throwing out individuals, throwing out loci, or in setting up experimental design. One of the problems is that just with cost effectiveness, people like now to get fewer individuals because it's often very logistically difficult, expensive, time consuming to collect individuals, meaning permits and everything. It's easier to just go in the lab and now tell somebody to do 10,000 or 10 million loci or whole genome sequencing. But the marginal benefits of doing that for any estimation can be generally pretty limited. So think carefully about experimental design. Individuals really valuable. This just shows how the changes in marginal utility translate into confidence intervals. So this is from the paper we did a few years ago. But it shows if you did parametric confidence intervals, assume that in this case, every pair of loci, or here for FST, every locus, and provides totally independent information. We know that's not true. If it were true, you would basically know the answer with almost no uncertainty after you had a few thousand loci. But these are the realistic confidence intervals based on, after accounting for the really high degree of pseudo-replication. But you can see that the difference is not as dramatic with FST. Again, you have a different scale of number of loci. So for the temporal method, relatively more useful to use large data sets. I think I may just stop here. I'm going to talk a little bit about setting confidence intervals. We're almost on to time, so I only had a couple more slides, and leave a little time for questions. One thing I'd like to say, I had references or to a bunch of studies that showed either talked about the issues on some of the slides or sources of some of the data. I'm working on a PDF document that I'd like to make available to folks that has all the references if people want to try to track them down. So I'll send it to the organizers, hopefully within a day or two, so they can. I suppose there'll be maybe an email list of attendees that you could share things with.
We can do that. Happily to do that. Thank you very much, Robin.
Yeah.
This was awesome. Are there questions?
Yes, thank you very much. It's very interesting aspects that you bring up here. I was wondering about this noise term, depending on the sample size of individuals. Do you still see this noise effect if you sample all individuals in a population, even if the population is like small?
Yeah. So the term I showed 1/S generally is assuming basically binomial sampling error. If you have actually sampled the whole population, then or say half the population, you could, I think, in theory, use a hypergeometric sampling variance, which is smaller. So the error term could be smaller. If you've sampled the entire population, let's see, for the temporal method, I think if you did that in a computer where you were sampling actual number of individuals and you sampled the reproductive parents, I think that might apply. But in, say, for the LD method, the theory for the drift term in the LD method is that you have sampled a infinite number of offspring, and then the 1/S term disappears. That's in Weir and Hill's paper from 1980. So if you've sampled all of the offspring for 1 year, yeah, I have to think a bit about it more, but it is an interesting question as to whether you have maybe sampled all of the individuals, but not all of the loci. Yeah, so it's getting complicated because those terms interact. They're shown separately there, but they actually interact because there should be a 1/S times any term. So it's a little more complicated than simply pretending you've got one of them eliminated. But it's an interesting question.
I have a hopefully an easier question. I just didn't understand the formula with the full SIB, half SIB. So you estimate any as 4 divided by something with half SIB and then full SIB. But I didn't understand what is it the number of half SIB? Is it?
Oh, I see. Yeah, that was Wang's. Let's see if I can find it. I think you're talking about Wang's, this one.
Yeah.
So, yeah, I should have explained that. No, this is the fraction. So you're getting a bunch of offspring, let's say 100 offspring, and you're trying to estimate the effective number of parents that produce them. So 100 offspring, you're going to do all the pairwise comparisons of 100 offspring, and you're going to put each of them each of the pairs in one of these three piles. Okay, and then you add up the number of full SIB comparison matches that you found and divide by the total number of comparisons. And that's what FS is. It's the fraction of comparisons that turn out to be full SIB. And it's 2 here because full SIBs give 2 doses of SIB chip. Half SIBs give 1 dose each pair. And for 100 offspring, 100 times 99 over 2 pairwise comparisons. That's basically 5,000, just under 5,000, I think. So then the number of half SIB you found divided by 5,000 would be FS, HS. Yeah. So that's what that is. Yep. Yeah. There's one back in the back, Alex.
Yes. Thank you. I just wanted to follow up on your comment about the estimate of any being downward biased when there's random samples of parents and offspring, but the ratio of any to n being unbiased. I'm trying to understand why that was. Is it because effectively, if you're assuming a random sample of parents and offspring, then you're assuming that your estimate of n is downward biased because you've missed the rest? Or is it because the estimate of NE is fundamentally downwardly biased, for example, because the ratio of the variance in reproductive success to the mean is expected to be downwardly biased when you've got a subsample of the population?
Yeah, so let's see if I can. I think that was further up here. Is the one you're talking about?
Yeah.
And so you're talking about this one or this one?
The bottom one.
Yeah, the bottom one. So the first part, if you have random samples of parents and offspring, you only sampled some of the parents, you don't have any information about contributions of some of the parents to offspring. Remember, the formulas for effective size have n in the numerator, so n is going to be smaller then. So NE, all else being equal, NE will be smaller. That's the source of the downward bias here.
Right. So it's a bias in
So
n, it's not a bias.
it's the
expected
bottom-
bias in the variance of the denominator part with the variance in k over the mean k.
Yeah, so the bottom part, yeah, so you have, it's not necessarily, yeah, you won't necessarily have any bias to the variance for the individual's parents that you have data for, if it's a random sample. If it's just the parents that are the
successful one.
poorest producers that you sampled, then you're going to have a bias in the mean and the variance. But if you have a random sample of parents that you have data for and a subset of the offspring that can be assigned to those parents, then the variance estimate should not be biased, I think. Yeah.
Thank you.
Yeah.
Questions? Yeah. Don?
Thank you so much for the presentation. It's really clear. And thank you so much for the presentation. It's very clear. I very enjoy. I have a question about what do you think about using POSEC with ALD?
Using what?
POSEC data, because calculate the R square, like how to make it possible to with POSEC.
Oh, sorry, you saying
POSEC sequence.
protest, coach?
POSEC sequencing.
Sorry, I'm not getting, what's the word?
It's like you pool, POLS.
Oh, pool data?
Pool data, yes.
Okay, pooled for individuals?
Yeah, and then sequencing them together.
Yeah, okay. Yeah, so so that's if the DNA content of each individual sample is low, and so yeah, it's hard to imagine how you could get useful data for LD from pooled samples of individuals.
So the main issue for the LD is this because the error is too huge? And
No, it's not that you really need to do LD, you need to have multi-individual for individuals, you need to have multi-locus genotypes
Yeah.
for individuals, not groups of individuals. So it's hard to imagine how you could get useful LD data from pooled samples. You might easily be able to get allele frequencies from pooled samples. So you could do FST or things like that.
Yep.
Potentially the temporal method.
Yep.
I don't see how you do the LD method. But could you do the SIBSHIP method? And I'm struggling to see how you could do the SIBSHIP method either with pooled data.
okay.
Yeah.
Yep, thanks.
Oh, there's another question. We'll switch maybe online.
Thank you. Okay, so this could be a bit of a risky question. You've made it very clear that NE is not NB, right? But still, on some of your slides, it looks like there's a simple relationship between NE and NB, like the number of breeders, right? So is it still a useful idea maybe to communicate to the layperson what NE actually is, to talk about, well, NE is not the number of breeders, but it's related or proportional to it under some conditions? Is that still useful to do?
Yeah, so I think, yeah, both these effective size ratios are potentially potentially very useful and potentially can be misused, both. So NE to N ratio, lots of people have tried to focus on it for obvious reasons. If it's easier to estimate one than the other, then you're more interested in census size, but you can only estimate effective size. If you know the ratio, you could expand up the census size or vice versa for effective size. The question is that these ratios are not invariant across species or even necessarily within species. There are quite a few empirical studies where people within a species, they have looked at estimated NE to N, for example, across multiple time periods. And one common finding is that many of these have reported an increase in the ratio of NE to N as N gets smaller. So at low density, estimates of NE to N often go up. So a possible explanation based on what term it's called, reproductive compensation, might be that, well, there's a lot of competition for mates at high density, and a lot of individuals don't get to reproduce. The wimps get pushed to the side and so on. So they end up being zeros in terms of reproductive success. But at low density, pretty much even the wimps are successful in getting a mate. So that's certainly a possibility. You have to be careful though, because there's also, when you're plotting, doing a plot, say, of NE to N versus N, there are statistical artifacts just in the fact that you're plotting those, you have one term, N is in both axes. And it can create, just with random data, it can create exactly that sort of pattern. So that's a caveat for that. For NB versus NE, that NE is generally going to be less than N. It's really kind of unusual circumstances for, at least for discrete generations in most species, for NE to be greater than N. But NB and NE, I always assumed going into the analyses of that NB had to be less than NE. It kind of makes sense. NB is just 1 year, and E is for a whole generation. That certainly was true with Pacific salmon. There are some pairs, they only reproduce once, so you've got different individuals across a generation. It makes sense, they're kind of additive, just like the seed banks. But for iteropara species, it turns out it's not necessarily the case. It's quite possible for NB to be greater than NE per generation. So there's quite a range across species. But the ratio turns out to be explainable or predictable just based on a few simple life history traits. Two most important being age of maturity and adult lifespan. And we have a paper that tries to explain, why that is. So within a species, based on certain life history traits, those ratios can be fairly predictable and meaningful. But they're not universally applicable across species that have different life histories, for example.
We are a bit behind schedule, but Anja, do we have maybe one short question online?
I think we have to address Joachim's point from the first part because we promised that. So that's again about the NE greater than 5,000 and the work by Lynch and Landy going back to that. So Joachim Merrier, he says, I think that Lynch and Landy made a mistake when assessing NE greater than 5,000 to avoid mutational meltdown. They modeled this with a mutation rate occurring in the effective size. So they considered implicitly that NE equals NC and assumed that the time an allele segregates only depends on NE and not on NC. So Joachim, not me, I showed recently that the ratio of NE to NC strongly influences the time to segregation. The paper about that is also in the chat for those of you who are online. And so he says, if you take that into account, the NE required to avoid mutational meltdown is typically 10 times smaller in real populations. So that brings back their calculations to NE having to be larger than 500.
Okay, that's great. I guess that's not a question, but it's very useful information. So with that issue, so it sounds like there's some recent work someone has done. Like I say, the last time I heard much about this was decades ago. So it's interesting to see somebody's worked on this more recently. And so maybe it'd be useful if that information could be made available, the publication or the reference.
Yes, for sure.
Distributed.